again, this is Matthew Dodd and welcome back to my YouTube channel. This has been the first video in a while. So this is going to be the first video in a short video series on bacteriophages. So in this video we're going to go into a brief introduction to the topic, very brief, and a brief history as well. In future videos we're going to go into some of the past, current and therapeutic and future therapeutic uses of bacteriophages, and we're going to explore possible uses and something of the relationship between viruses and bacteria, which can be characterized by bacteriophages, and some of the unique mechanisms and processes that bacteriophages have exposed. So geneticists like Max Delbruck utilizing bacteriophages to discover new things about biology. I mean, bacteriophages being a catalyst for scientific discovery all the way back to the days of Durrell and Bordet, which we're going to be looking at today. I hope you enjoy. If you want to, please consider dropping a like, subscribing, or checking out some of my other content. And yeah, enjoy the video. Thank you very much. So, what is a bacteriophage? So bacteriophages are viruses that specifically target bacterial organisms. So it's important that viruses are incredibly specific in their targeting, often down to very specific species, which is why a big deal is made out of whenever a virus will transcend the boundaries of an animal species, for example bird flu, and achieve zoonosis, transmission to humans. But typically viruses are incredibly specific. And that's because they have spike proteins which allow them to invade very specific cell types. So there are a lot of viruses. 10 to the 31 bacteriophage particles exist on Earth. So around 1 trillion per grain of sand, with tens of millions of species as well, so a lot of viruses. So let's define our terms. So viruses are non-living particles which mimic some of the processes of cellular life. So they reproduce, they move, but they're not living. They're deemed the ultimate parasites. Obligate parasites is what they are, which means that as a necessary and ubiquitous function of their existence, they parasitize other organisms. So they hijack host cell machinery, inserting their genetic information into the genetic information of the host cell. So that might be a plasmid for a bacteria. For human beings, it will be chromosomal DNA. But they'll hijack the DNA of the cell and use the cell's machinery and infrastructure to create multiple copies of themselves, which the cell will then be lysed, releasing viruses. So there's many problems for the organism in question along that road, whether the organism is eukaryotic or whether indeed the entire organism has been killed by the virus splitting it open in that sense. But yeah, they're obligate parasites and quite interesting organisms with a lot of potential, and also, as we've seen recently, a lot of potential to do quite devastating damage. But yeah, bacteriophages can be defined as viruses that attack bacteria and are very selective for specific types of bacteria. So I'm going to briefly foray into some very basic bacteriophage characteristics. So as you can see on the slide behind me, there are a vast range of morphologies. For example, coronavirus resembles a laundry ball, if you've ever seen a laundry ball with little spikes on it. But there are plenty of other morphologies as well. So for example, a typical bacteriophage morphology will have an isohedral capsid at the top, which will contain the genetic material. There will be a tail tube with a sheath attached to a base plate with tail fiber protrusions. And the overall effect is the bacteriophage actually looks quite like an alien walker from something like War of the Worlds. So they're quite an intimidating particle. Though, yeah, not super dangerous to us, thank goodness. Thanks to viral specificity, which will be an interesting point for later videos, since viral specificity might actually prove quite a useful tool for treating cancers, among other things. Yeah, so as you can see, viral bacteriophage morphologies differ greatly from capsid size and the number of polyhedral faces, tail tube, length, protein composition, and quite importantly, the composition of the genetic material. So in this slide, you can see some bacteriophage categorizations. So categorizing bacteriophages by their genetic material, or indeed viruses, is known as the Baltimore classification, which is a bit old, but still, in my opinion, somewhat useful. And yeah, you can have single-stranded DNA, single-stranded RNA, double-stranded DNA and RNA. There are different genetic materials viruses can carry around, and upon them has been exerted selective pressure. So whichever genetic material is most metabolically effective for the viruses will probably be the type adopted by species that target an organism, and that varies a lot based upon a very vast array of different environmental and organismal factors, which we won't quite get into in this video, thank goodness. But yeah, some viruses like the HIV virus is a retrovirus, which is single-stranded RNA, and it will require a reverse transcriptase enzyme to convert its retroviral RNA into DNA, which is of course the genetic material it would require to insert into, say, a bacterial plasmid. But yeah, so that's bacteriophages, a very basic categorization and morphological overview. I hope that's been illuminating. And now we're going to get into the history of bacteriophage discovery. History of bacteriophages. So as you can see behind me on the screen, I've got a brief timeline of different points in the early history of bacteriophages for this half of the history of bacteriophages video. We're going to start off with Frederick Tort, who was growing micrococci, yellow and white, as a byproduct of trying to isolate and propagate vaccinia virus. He was interested and quite successful in propagating colonies of viruses in order to make vaccine preparations. If you'll remember, back in this time, this is where we've got basic vaccine methodology emerging, where, for example, cowpox samples are being created and propagated and injected into individuals in order to hopefully, and quite effectively as it would turn out, engender immunity against the more deadly smallpox. But anyway, that's what Frederick Tort was doing. He was trying to grow vaccinia virus. He was growing cocci, and then he noticed that in his preparations, the cocci were dying in small holes. So there would be little circular diffusions in which these bacteria he was growing would die. So he didn't quite know why this was. He determined that it may be an error. But one of the conclusions he drew was that there may be an ultramicroscopic virus. Frederick Tort was the first person to establish this phenomena of bacteria dying in this way and publish a paper on it. So next you've got Felix Terrell, who was a very interesting character and influential figure in the history of biology and biochemistry. So a man who would work as a surgeon and doctor in wartime and make numerous trips around the world and make many different observations. So in 1915, the same time that Tort published his paper, Terrell alleges that he came up with the idea of bacteriophage, but this is where he first coined the term. Well, later in 1917, Terrell would publish his first paper where he talked about bacteriophages as individual propagating organisms, though his conception of bacteriophages was slightly different to what we now understand them to be. He thought they were some kind of immune defense mechanism within the body that were kept in the gut. I mean, he also believed that distinctions between bacteriophages were phenotypic rather than different species, as in there was one universal bacteriophage species utilized by the human gut in order to kill harmful bacteria, and that they would phenotypically express specificity for bacteria. However, his theories were influential and a good step in the right direction, which we can say in hindsight. However, they were fiercely criticized by Jules Bordet, who we can see on the timeline. So Jules Bordet would oppose this very harshly. He had a theory of autolysis, so he thought that this was a transmissible gene that bacteria would acquire via Lamarckian theory of evolution. So he was of the Lamarckian school, which has been since discredited, but that's an interesting rabbit hole if anyone wants to go down that. He thought that bacteria acquired characteristics which incurred them to autophage. So they would destroy themselves, and this was something they would pass on to each other. And that this kind of genetic material that they'd pass to each other was a sort of illusion that... So one of the experiments that he would use to try and prove this was hypothesizing that if you had an illusion of dead bacteria, that it would contain a certain concentration of the substance, the autophagic substance that bacteria would induce and thus destroy themselves, so that if you incurred serial dilutions, you would see an incremental decrease in the effect. So that was one of the experiments he ran. This experiment was run by multiple different people, and people would interpret the results differently as bacteriophage versus autolysis. I've got as an arrow on the timeline. This debate went on for a very long time, and so different scientists would take different sides. So, for example, American biochemist John Howard Northrop would support Bordet and base his interpretation on bacteriophage kinetics, and enzyme accumulation, which was his interpretation of the autolysis theory, autocatalytic enzyme activation. So yeah, the scientific community generally agreed that with Bordet and Northrop and their ilk, Bordet was a Nobel Prize winner, and these were very influential scientists, whereas Durrell didn't quite have the academic chops of his peers without a formal university level education, as I understand it. But, well, he would be proven to be right in the end. He had some more high profile allies, of course, in this bacteriophage versus autolysis war. So one of them would be Belgian scientist Richard, I'm going to butcher this pronunciation, Bruynog, who was director of the Institute of Bacteriology in Leuven, a city located near Brussels. So he was the head of the Pasteur Institute, and he backed the bacteriophage theory and encouraged a lot of his students to do the same. So a lot of students, Belgian specifically, as the Springer Link article outlines, would get into bacteriophages as a consequence of this quite high-profile debate that went on in the community, which in a sense you could almost say was a proxy for the scientific debate around Lamarckian versus Darwinian evolution. But again, that's a different rabbit hole. But Bruynog would make several papers. He would try and persuade Bordet. The limiting dilution technique, Appelman's phage titration, and one of his students, Rien Appelmans, after which the Appelman's phage titration is named, would try and persuade Bordet, but Bordet wouldn't have it. So yeah, this was a very long-winded debate, which was only really conclusively decided, unbeknownst to most of the scientific community due to the circumstances of the time, when the Ruska brothers in Nazi Germany would first take photos of the of bacteriophage particles. So on the timeline it's a bit faint, but you can see that there's little particles surrounding what are pictures of electron microscope pictures of bacterial molecules, and these are the first pictures of bacteriophages that were created. So the last little thing I've got on this timeline is the Phage Treaty, and the Phage Treaty was made in 1944, just a treaty of research and expansion into phage research. I mean, of course, in the West, the idea of phage therapy to treat bacterial infections had been mostly thrown out in favor of penicillin and other antibiotics, which were miraculously discovered by Ian Fleming and others. But the Phage group would still go on to make many important discoveries in the fields. are in the field of molecular biology. So, of course, the most influential member would be Max Delbruck, and he would form the Phage group. So Delbruck, Salvador, Edward Luria were the first three members, and they would be awarded a Nobel Prize later. But yeah, there's a great deal of bacteriophage research and with a great deal of implications on molecular biology which would come as a result of their work. But yeah, that's the first section on history of bacteriophages. Thank you very much. So thank you for watching this video. I hope you enjoyed it. I'll shortly be publishing the next video to outline the second half of the history of bacteriophages. I'm going to try and keep these videos a bit shorter, between 10 and 15 minutes in length, which maybe floats your boat, I don't know. Please leave in the comments if there's any comments you'd like to make on structure, length of the video, anything you learned, anything you'd like to correct or improve. And yeah, thank you for watching. Have a great day. Consider subscribing, liking, and checking out some of my other content like my Aducanumab series. And yeah, have a good one. I'll see you all soon.